So today I'm going to tell you about this an approach to differential expression you might like to think about. So it's a method that we've developed in the lab for examining differential expression. You might like to think about gene expression. And you can read about the paper. I've just added the reference to the paper. This was published this year, so you can look up the paper for more details. So we called it the characteristic direction. It'll become clear why we've called it that. So first I want to make the point that what we've made is it might be if you're coming from a certain perspective, you might think of it as just a technicality of a bit of data processing that we're doing that's sort of a kind of a side issue. But it's actually it's very important. There's a number of issues that we have to address if we're going to make meaningful biological inferences from these genome-wide data sets. So I'll skip over that point, but there you go. So I'm going to show you our approach. With this approach that we've developed in the lab to overcome some of the issues involved in differential expression analysis. And then, I'll describe the approach and sort of kind of justify it sort of conceptually, and then I'll show you how it. I'll kind of benchmark it, and I'll compare it to other approaches. Hopefully, you'll be convinced that it's quite a lot better than at least the ones that we've compared it to, which are kind of popular, often used approaches. And then, if I have time at the end, I'll go into sort of a natural extension of this approach. So hopefully, I'll show you it's kind of it gives a nice sort of geometrical, visualizable image of differential expression analysis, and it leads to some natural generalizations. And I'll show you one for genes, the enrichment of gene sets. So this is kind of how differential expression analysis is going now. It's going from focus is changing from the level of which genes are differentially expressed to which collections of genes are differentially expressed. So this approach naturally sort of develops into that. So first, I just quickly sort of give a sketch of some of the challenges that we're trying to address with this method that we developed. So the first important point is that when you think about gene expression, you're looking at the expression across the whole genome of many genes, and they're not just all acting independently, they're actually all talking to each other in this complex network of interactions. And it's important that I think it's important that your analysis sort of bears that in mind, or it takes that into account. It's sort of important statistically. So let me give you an example of what I mean by this. So let's think of a gene expression profile that consisting of just two genes, gene 1 and gene 2. Then one gene expression profile would then be represented as a point in the space, as we did in principal component analysis. Other talks that I might have given. So with one sample, we have limited information, but there's going to be some natural variability. The expression of these genes are going to vary, not just because of noise, but because of certain natural biological processes. And so as you collect more samples, you get more points, and you might see patterns developing in the space as you get more samples. And what we're going to try and do is we're going to try and take advantage of these patterns and the structures of the natural variability in gene expression space in order to get a clearer picture of differential expression. Typically, like the most common methods, they ignore this kind of this multivariate structure, and they just look at each gene separately. This is sort of good, sort of conceptually and sort of calculation, like computationally, it has some advantages to do that. But you're losing. I think in our method we argue that you're losing something important when you do that. You're missing out on the natural variability and the fact that genes are talking to each other. These are very important in biology, and we try to use them. A second issue I'll not spend so much time on, but it's quite important, which is something that somebody called it the curse of dimensionality. So that's not really apparent in this picture because we've only got 2 genes, so we don't need very many samples before you can sort of see the structure, this structure that's appearing, which is a simple correlation between the 2 genes. But if you imagine, if instead of 2 genes you have 20,000 genes, these points become sort of, the space sort of increases in its dimension, and these points become sort of very dilute. And so, to get the same sort of density of points, you need exponentially more samples, which you don't have. You never have as many microarray profiles as you do genes, and this is a statistical issue, and we deal with it in a way that I'll show you. So those are a couple of the issues that we try to address with this method. Bless you.
Thank you.
Okay. So I'll go into a bit more detail about this one, which is kind of the main point that we're trying to address, which is this multivariate versus univariate approach. And here with this picture, I've got a sort of a visual example of why taking a multivariate approach might be better. So let's think. As we said, genes are talking to each other. One very simple way that 2 genes can talk to each other is they could simply be correlated in their expression. So the expression of gene 1 goes up, the expression of gene 2 tends to go up. It's a very simple communication between genes. Very simple network there. Now, let's now examine some differential expression. So we've got two classes of samples, like diseased and normal, or perturbed against drug-treated cells or versus control. And we want to look at how does the genome-wide expression profile change between these 2 classes. So I've colored the 2 classes red and blue. Now, with a univariate approach, we would look at gene 1, and then we'd look at gene 2, and we'd say, okay, is gene 1 differentially expressed? Is gene 2 differentially expressed? So let's try that here in this example. To look at the expression of gene 1, just let all these points just fall down onto this horizontal, and then look at their distribution. So these curves here are the distributions of the samples, of the expressions of gene 1. And what you see is there's quite a lot of overlap. There's gene 1. If you look at those distributions, there's such an overlap that you might say that gene 1 is not really differentially expressed here. As a t-test might say that's not a very significant difference. And you can look at gene 2. You can let all the points fall that way onto the vertical, and look at the distribution. And again, you see there's such a large overlap. You might say no, gene 2 is not really differentially expressed. But if you look at the whole picture, just look at it now, all together, gene 1 and gene 2 together, you can see very clearly there is a very clear difference between the red points and the blue points. They're very clearly separable. But you only see that so clearly when you're looking at everything together. You're looking at the 2 genes together, not just looking at one and then the other. So this is the first point that we would like to make. If you multivariate approaches in conjunction with natural biological variability and networks of interactions between genes is a better way to go with differential expression. And then to get a bit more specifically into our approach to this, we're going to we're going to say, okay, if we look in this direction, we don't see much differential expression. If we look in this direction, we don't see differential expression. But if we pick a direction very carefully, this one, and let the points fall on this direction, then we see a very clear separation. So this is going to be, sort of start to lead into our approach here, is to, how do we identify this direction, a single direction in the expression space, which is best to look in if you want to separate between the two samples. And with this, we're going to use this direction to characterize the differential expression, hence the name, the characteristic direction. So I'll go a bit more specifically. You don't need to worry too much about the specifics, but this is just to sort of give you a flavour of how we calculate this. It's done using a method of classification called linear discriminant analysis. And what this is a kind of analysis that what it tries to do is to give you a classification, so that if you give this analysis a gene expression profile, the classifier will tell you, I think this is a normal sample, or I think this is a diseased sample. So it's a way of dividing the space up into normal and diseased, based on what you know. So let's say that here's our data. We've got red and blue samples, and we've got two genes. The first thing we do with this analysis is we're going to model it with a normal distribution, but not just an ordinary one-dimensional normal distribution, a two-dimensional Gaussian distribution. So it's defined with a mean vector, which is the mean of x1 and the mean of x2, and a covariance matrix, which is composed of, in this case, it's two-dimensional. We've got the diagonal is composed of the variance of each gene, and the off-diagonals is the pairwise covariances between gene 1 and gene 2. So the covariance matrix sort of tells you in what direction the distribution is sort of stretched. Okay, so we can estimate this covariance matrix just from our data. We can calculate the estimators of the covariances and the variances of the data and the means. The next thing that you do in this analysis is that you divide the space into regions that are more likely to be red samples and more likely to be blue samples. So all you do is it's just based in terms of those Gaussian distributions, their probability distributions. Wherever the red probability distribution is greater than the blue, you color it's colored red, and it's considered more likely to be red. And then there's a boundary between the two. This is it's what's called the separating hyperplane. Now in this case it's just a line. You might not think it's a plane, but in higher dimensions, so in 3 dimensions it would then become a plane, and then in higher dimensions it would be what we call a hyperplane. But it's an analogous concept. Now, so this is the plane which best separates the two, the red and blue samples. Now, so it's sort of a geometrical fact that a hyperplane has an orientation. You can imagine tilting the hyperplane in different ways, and the orientation is defined in terms of a single vector, the vector which is normal to its surface. It's at 90 degrees to its surface. This one here we call B. So this is the direction. This is what we're going to call the characteristic direction. It's the direction which best characterizes the difference between the red and the blue samples. And it's calculated, in terms of this analysis. It's calculated in terms of the mean of your data and the covariance matrix of your data. And it's quite a simple calculation. But that's the main point. You don't need to bother with all this detail. It's just that's the concept, and it's quite simple to calculate this. But what you get is a vector, a direction. So the next thing we're going to want to do is we want to interpret this. We've got this vector, and it's characterizing our differential expression, and now we want to interpret it. Like, okay, all right, great, we've got a direction. What does that mean for the genes? Which is the most important gene? Which is the least important gene? Now, the way we do this is very simple. In this case, in this two-dimensional case, this direction can be aligned more or less with the coordinate axes corresponding to each gene. So if the direction is pointing more towards more aligned with this axis, then that means gene 1 is more important. If the vector is more aligned with this axis, then gene 2 is more important. Another way to think about it would be in terms of a sort of a Pythagoras. So we have a right angled triangle here, and the components of the vector are the sides of this triangle, and then the sum of the squares, so this kind of the magnitude of these components, add up to some number, which we always normalize to 1. So what we're considering is not the size of this vector. We don't care about that. We care about the direction that it's pointing in. So this is always sort of scaled to 1. So this value is always 1, and then the sum. This is the sum of all the components. And so what we interpret for each gene has a corresponding component, and it contributes to this sum. And the genes that have the biggest squared component contribute most to this sum, and they are then considered the most important for the differential expression. So this is how we interpret the components of this vector. So the next, that's how we deal with this issue of multivariate versus univariate. The next sort of issue we wanted to deal with was this curse of dimensionality. So if I just skip back here, we're calculating this direction by multiplying the difference between the means by the inverse of this matrix. Now this is the covariance matrix. It's got dimensions. It's a square matrix with the dimensions equal to the number of genes by the number of genes, and that's we've got components. So if the number of genes is n, we've got n squared variables in there, or half that because there is a symmetry. But even so, that's a lot of variables that we're estimating. We're estimating a lot of covariances from just relatively few samples. So this is a statistical issue. We're estimating a lot of things from a few things. So we expect to have lots of errors, uncertainty. So this is how the curse of dimensionality manifests in this problem. So essentially what we do, we use a method which is sort of very commonly used, called regularization or shrinkage. And what this essentially does is it takes the covariance matrix and it kind of smooths it. So we're expecting lots of noise, but what we want to do is we want to smooth away the noise while preserving some, the signal as best we can. So what we do is we perform a transformation on our covariance matrix, and we take our original, and then we take a kind of a smoothed version of it, this diagonal version. So we kind of we do what's called shrinking towards the diagonal, and this involves a shrinkage parameter which tells you how much smoothing you've applied, and you can use something like cross-validation to choose the value of this parameter. All right, so that's the method. That's the concept of the characteristic direction. So what I'm going to do next is I'm going to sort of benchmark it and show you at work, and I'm going to compare it to these methods here, because these are sort of quite commonly used approaches to differential expression. So one of the first methods to be used was simply fold change. It's just taking the ratio of the expression of each gene separately. And so the genes with the biggest fold change would be considered to be the most important genes. A slightly more sophisticated approach would be to use something like a t-test. Now, in the t-test, you're starting to use some information about the variance. How is each gene varying? You estimate the variance of the gene to perform the t-test. Is the difference in expression large compared to the variance of the genes? But still, you're working univariately. And there are two other methods that are a bit more detailed, which you'll find in like bioconductor or things like this. They're used very commonly. Significance analysis of microarrays and LEMA. you can read about them if you like. These are like perhaps the most commonly used methods. So now, how are we going to decide which one of these methods is doing the best job? It's kind of tricky, because typically with differential expression you don't know what the right answer is. And in the case where, if you do apply it to synthetic data where you know what the right answer is because you input the right answer, you're missing out on all the sort of the details of the structure, important structure that you find in real data. So the approach that we took to this was to use differential expression data. So we've got gene expression data from control situation and a perturbation of a single transcription factor. So for example, a knockout of one transcription factor. And then, we're going to examine the differential expression. So now we still don't know the right answer, but we can get a clue to what the answer might be using an independent data set. We use ChIP-seq data for that very same transcription factor. We look at the ChIP-seq data of where does it bind on the DNA, and then we identify the genes that have a binding site in their promoter. So then we get a subset of genes which have a binding site for the transcription factor in their promoter, and then so it's not necessarily true that all those genes will definitely be differentially expressed, but we might say that they're more likely than most genes to be differentially expressed. So this is our approach. We've got a suggestion of what the right answer is. We've got a kind of instead of a gold standard, we've got a silver standard. And so our approach is to take our gene expression data, so genes down the rows, samples across the columns, control here, and transcription factor knockout here. And then we're going to use each of the methods in turn to rank the genes. So we'll put the most significant gene on the top and the least significant gene on the bottom. Each of these methods has its own perspective on what that is. So then the next step is, for this transcription factor that was knocked out, find those genes that have got a binding site in their promoter and see where they sit in this ranked list of genes. So I've highlighted them red and put a little arrow up here to illustrate. Now, if our differential expression analysis is to simply randomly pick genes, we just put them in a hat, then what will happen? What will this look like? The red arrows, the red lines will be sort of uniformly spread throughout this list. They'll be just like a completely random. But if we do a really careful job with the differential expression analysis, what we might hope is that the genes, these red genes, the ones with the arrows, will tend to be at the top. They'll tend to be found to be differentially expressed because they have a binding site for that transcription factor in their promoter. So what we're going to do is we're going to take this list, we're going to lie it down flat like that, so the significant genes here, least significant genes here. Then we're going to walk down the list and keep a track of what fraction of all of these genes we've discovered as we walk down. Now, if we just picked randomly out of a hat, we'll expect sort of like a constant rate. It'll be just this dotted line would be just the, what you'd get if you just completely guessed. But if you tend to be identifying these silver standard genes, then they're going to be concentration of them up here, and you're going to get a cumulative excuse me, distribution that looks like this. So then let's subtract the random part, because we're not interested in the random part. We just subtract the dotted line from the filled line, and then we're going to get a curve that looks like this. And then what we're going to do is we're going to take the height of that curve as a measure of how good your method of differential expression analysis is. It's a measure of how much have you prioritized the correct genes. So here's the results. So we did this over 73 transcription factor perturbations, and this is kind of like an accumulation of all that. So each one of these is one of these curves, but it's actually done for real data and for each one of these methods. So what you can see is, okay, green one, fold change is doing nothing. It's just a flat line. It's basically like random almost. And then where's these others? This dotted line, lemma, is a really popular method. It's doing something. It's prioritizing the genes. And then here is the characteristic direction. This one. So it's to a much greater extent, it's prioritizing the so-called correct genes. Each one, there's two pictures here because we choose two different databases of ChIP-seq data. We've got similar results.
Oh, this approach is multivariate approach when you are comparing.
Yes. Yes, so each one of these curves is the result of applying one method of differential expression analysis, and it's sort of evaluating to what extent do you prioritize the genes that have a transcription factor perturbation in their binding site under perturbation of that transcription factor. So, if you look with the characteristic direction, the method that I sort of described, then you prioritize the correct genes much more highly than in these other methods. So our interpretation of this is that characteristic direction is a better approach to differential expression. That would be the most obvious interpretation of this. Here we tried it in a completely different setting. We looked at not transcription factor perturbations, but drug perturbations. So that's on the left here. So we've got some cells that are perturbed with the drug, and then we try to see, are genes which code for proteins which are putative targets for this drug, are they differentially expressed according to these different methods? And you get some wiggly lines. The answer seems to be no. But if you then go to the protein-protein interaction network and look for neighbors of those, so these are genes which encode for proteins which interact with the drug targets, then you start to see a signal again. And the pattern looks very similar. Characteristic direction is again prioritizing these so-called, these hypothetical correct genes much more highly than these other methods. So that's another sort of, it helps reinforce this idea that we're doing a good job. So this was all for microarray data. Lots of the research at the moment is now how do you treat these high throughput sequencing, because there are some issues that are sort of specific to that technology that a lot of papers are trying to sort of resolve at the moment. So we wanted to test whether this approach will also do well in this setting. So there's not so much data. The results I've shown you previously were over a very large number of data sets. Here there's not so much data. We picked one, which is from this paper here. And what they did is they have differential gene expression and also differential binding of STAT3. So that they're interested in this one particular gene. And so and they use this method called RNA-seq, which is currently the most popular method of looking at differential expression with RNA-seq data. And so they looked for consistency between, are genes which are differentially bound by STAT3 also differentially expressed by STAT3 under perturbation. And they find, so this is this purple curve here, they're finding, those genes which are differentially expressed tend to be those genes which are differentially bound by STAT3. But if you look with the characteristic direction, you see it's much more so, which means there's much more consistency between the ChIP-seq data and the RNA-seq data, apparent consistency when you use this method. So this is another, it adds to the confidence that this is doing something also in the setting of these high throughput sequencing methods. So we think that's the best way to sort of benchmark this data, using real data if it's possible. Reviewers wanted to see synthetic data, so we did it. We've made up we've made some imaginary gene expression data, and we've given it all the properties, as many of the properties of real gene expression data as we can. And we've done the same thing. These are essentially those same curves. We've defined certain genes to be differentially expressed, and then we try to find them. These are, using these different methods, and again, characteristic direction comes out on top. So one of the things that I like about the characteristic direction is that it gives you a sort of a nice natural picture. You can think of, you perturb some cells, and you think of them in gene expression space being pushed, from this state, pushed into that state in that direction. And it's kind of like and that gives you a nice picture. And this picture can sort of, it can be taken further if you think of a pair of perturbations. A natural sort of concept for their similarity is this is the kind of the angle between these two directions. The more aligned two perturbation directions are, the more similar they are. And there's also this that gives you a nice picture of what gene set enrichment might mean in terms of these geometrical concepts. So here's the characteristic direction, this arrow here. Here we've got 3 genes now. We're thinking in 3-dimensional space. And let's say we're interested in a gene set that's composed of gene 1 and gene 2. And we want to know, is gene 1 and gene 2, the gene set, is that collectively differentially expressed? Now, gene 1 and gene 2, they define a plane. This plane that is spanned by those 2 coordinates. So you can calculate the angle between the characteristic direction and that plane. And the smaller that angle, the more aligned the direction is with this space that is spanned by a gene set, then this is a kind of a quantification of enrichment. So we've developed this approach. We've applied it on some data. We've got 8 data sets where we've got 2 classes of samples, one from young humans, one from relatively old humans. But they're otherwise similar. And then we bring in a gene set library, which is composed. It comes from this MGI mouse phenotype library. So these are genes that are sort of associated with a phenotype. I think there are 472 phenotype terms, each with a list of genes which, when knocked out, sort of cause that phenotype. Here's an example of a typical line from the ontology. You've got the code, you've got a phenotype, and you've got a bunch of genes that are associated with that phenotype. So we took these data sets and we looked, for example, here we've got phenotypes that are associated, that are sort of related to morphology, processes with morphology, and we see no signal in any of the methods. And the same for development and behavior. But if the phenotype terms that are associated with aging, like premature death and so on, with this principal angle enrichment approach, we are seeing that we're identifying them as significantly enriched, whereas this is missed with other approaches. So this is kind of given us a clue. We're just finishing a paper actually where we've got more evidence that this is actually a better way to look at gene set enrichment analysis as well as sort of differential expression. Yeah, so that's it. So I tried to describe this new approach to differential expression and how it tries to tackle 2 important issues in differential expression. One, the univariate versus multivariate issue, and also the issue of the curse of dimensionality. And I've tried to demonstrate that tackling those issues actually leads to some significant improvements in your analysis. So this is good because you might, if you're doing a better job of the differential expression, you will hopefully be able to gain more biological insight from the data. And with this approach, it seems to be sort of naturally leading on to other sort of extensions. I didn't put the link, but there's a package, an R package that we've made, which you can get from the CRAN repository in the CRAN. It's called Geode, capital G E O D, capital D, capital E. So you can download that, and you can you can use this. This package performs the characteristic direction and also the enrichment analysis as well.